We researched the different types of search methods an investigator would use to search a crime scene. Some of the methods that we researched include the grid, spiral, quadrant, and wheel method.
Our second project was the Human System Body Project. In this project, we learned how the 11 body systems function, or 12 if you count the female and male reproductive system as different. We learned about the respiratory system, reproductive system, muscular system, cardiovascular system, skeletal system, endocrine system, lymphatic system, integumentary system, urinary system, nervous system, and digestive system. It was by no means simple to create a pamphlet for all of this to help us learn it, but it was very fun and very helpful. Creating it showed us where each part of each system was in the body, and also describing it in the back of the pamphlet helps us remember what each system does. This project was the DNA replication and RNA transcription or translation project. In this project, we learned about how DNA is replicated so mitosis can take place. We also learned that how DNA is transcribed into RNA, which is later translated into proteins, hence transcription and translation. We learned DNA replication by making a model of DNA itself and matching the correct base pairs with each other. We learned RNA transcription and translation because we would need it to dive into areas later into the year that we would need to know the RNA process for.
We did a heart dissection lab where we traced the flow of oxygenated and deoxygenated blood. We also used this lab to see the different structures and the functions of the heart. We learned how to find the calories in the food by using a calorimeter. We must follow precise steps and certain rules so that we can get an accurate answer. We also built our own calorimeter using tinfoil and a can, burning the food underneath it.
Understanding nutritional concepts, the nutrition labels, and research was an important part in the Year 1 biomedical course. We divulged into various nutritional concepts like cholesterol, fat, and vitamin types, which included their physiological function and relative healthiness to the body. From there, we learned what happens in types of diabetes and their causes. In the Year 1 biomedical course, we also became familiar with genetics. We were taught how to predict an offspring's genotype through monohybrid and dihybrid crosses. Different genotypes, such as sex-linked genotypes and multi-allele genotypes, were taught and expanded our understanding of genetics as well. In the entire year, learning about the cell membrane was among the most important topics we learned. We were taught the structure and function of the phospholipid bilayer of the cell membrane, including its individual phospholipids of hydrophilic glycerol heads and hydrophobic fatty acid tails. Later, we discussed cell membrane proteins, which included integral and peripheral proteins.
We researched and cataloged different viruses to learn more about their structures and characteristics. We also used our research to compare viruses and bacteria. We researched a specific virus and made a PowerPoint explaining its transmission, history, and other characteristics about that certain virus.
Our last project was our inoculation project, in which we learned how to use the antiseptic technique to grow bacteria in a petri dish. We learned this by making a tutorial on how to use the technique in a lab. At first it seemed very difficult, although with some guidance from our teacher, we were able to get it done quite easily.